Okay, thank you. Welcome. Thank you for this opportunity to present my research work here today. My name is Natalie Hirst and I'm from the Faculty of Biological Sciences, as Tony said. And my research is entitled Saving Lives, Early Detection of Cancer Surgery Complications at the Bedside. And this is supervised by Professor Milner, who's a bio-nanotechnologist here at the university, and Professor David James, who's a consultant colorectal surgeon here in Leeds. So I'm just going to start with a bit of background about my research in terms of defining the clinical problem that we're trying to solve. And this revolves around bowel surgery. Hopefully you can see these images. There's a picture of the bowel here, and you can see the bit that we're cutting out there. And we commonly cut out bits of bowel due to cancer, but might also be non-cancerous benign disease. So that's the bit that we chop out, and then what we do is we simply join the loose ends back together to restore continuity of the bowel and make a join, as you can see there. And thousands of patients get this kind of operation worldwide every day. When this joint leaks, it's a catastrophic surgical complication. The leak rate you can see there is between 1 and 30% of patients, depending on various risk factors. So up to a third of patients can be affected by this. The top two pictures, which haven't come out very well, and that might be a good thing, they're quite gory for anyone who faints, but they're the only ones I've got, I promise. Or what you typically see at reoperation for this joint leak problem, and that's one of the treatments that we have for it. I just wanted to really demonstrate how the bowel looks very stuck down together due to all this infection that's happening with this complication. The bottom picture is a patient on the intensive care unit where these patients invariably end up due to this infection that starts in the belly, then travelling around the body and starting to affect other organs and shut down other organs. So it really is quite serious. And having said that, yes, we know that the mortality rate, the death rate, is 7 times higher in patients who have a joint leak compared to those with a normal post-operative recovery. Your other complications are much higher, and even your risk of cancer coming back is higher if you have this joint leak, obviously if your original operation was for cancer. And this is all with a huge cost burden to the NHS. So hopefully I'm driving home the point this is a bad complication to have. Unfortunately, we're not very good at diagnosing it for a variety of reasons. Firstly, it has quite an insidious presentation, so patients can look very fit and well and strong in the days after their surgery. While they're leaking, then they suddenly are very, very unwell. It's obvious that they've been having this leak, but we haven't picked it up earlier to do anything about it. The current diagnostic tests that we have are also quite poor. We're limited to things like blood tests, so looking at markers in the blood, and as you can imagine, this is quite a late sign, again, after that infection's already travelled around the body in the blood. We can also do X-ray type testing, such as a CT scan, a radiological image, which tend to be better, but they have their own problems in terms of they're costly, they can be difficult to arrange, and we can't do them a repeated number of times on the same patient. And yet we know that the longer the delay in diagnosis of this joint leak, the worse all these complications get. So there's this real window of opportunity where if we can diagnose it earlier, we can treat it earlier, and minimise all the complications that I've talked about. So there's a need for an immediate specific diagnostic test for this joint leak, and we think a biosensor test might be the answer to this. So what do I mean by a biosensor? Hopefully this picture is fairly familiar to a lot of you. It's a glucose sensor, a glucose monitor, used by millions of diabetics around the world every day to monitor their blood sugar, and it's a biosensor. So the patient pricks their finger, they put a bit of blood on the top white disposable test strip you can see there, and the computer monitor gives them a number out, a meaningful number of what their blood sugar is for that day. But broadly speaking, this is what a biosensor is. So the white bar is your test strip, or in most cases an electrode, onto which you attach something that's going to bind with whatever you're trying to measure. So in this case the glucose there binds, and it generates generally an electrical type signal, is the best way of thinking of it. And this goes into the computerized system and again gives you out a meaningful figure immediately without any other equipment required. As I said, diabetics use their finger prick bloods, but we're looking at whether drain fluid is a better way of diagnosing the bowel joint complication that we're looking at. So when a patient has the operation I described, they wake up with a plastic drain tube coming out of their belly wall, which hopefully you can see in this picture. And it just drains into a bag next to them in the bed, and it drains all the effluent and fluid from around where we've been cutting and from around where we formed this joint that we're talking about. And so logically speaking, this should be a more sensitive early test because it's measuring things that are around that joint instead of things that are in the blood that will be later on that change. So what we're going to measure in the drain fluid, we're aiming to make a series of 3 biosensors to 3 different things that are changing in the drain fluid as this patient has a leak. So first of all, we'd like to measure lactate, and this is a sign of the initial ischemia or lack of blood supply to this joint, which is a very, very early change. We can also measure inflammatory cells, so as this infection in the belly progresses, the patient's own bodily defences start to kick in and increase white cell counts and inflammatory cells as well. Or we can measure bacteria. So there are normally bacteria within the bowel itself, but not outside of it in the abdominal cavity where this drain's sitting. If this joint leaks, the bacteria will leak out into the abdominal cavity, and we can hopefully pick them up in the drain fluid. So the first kind of biosensor that I've chosen to work on is to lactate. And this is a schematic of what it looks like in the lab, but there are the similar components to the glucose sensor that I showed you. So the black bar is our test strip, our electrode, onto which in this case I've attached an enzyme so that we can see the reaction. It's still hooked up to the computer type system, and it's all dipped in a big pot of initially buffer salt solutions for initial kind of testing, but ultimately our drain fluid samples that we're going to measure. And this is how it works. So we add some lactate, the enzyme catalyses a reaction to form amongst other products hydrogen peroxide. This undergoes its own reaction, which changes the number of electrons in the system, or changes the charge of the system if you like, and this is measured back at the electrode there. It's fed into the computer system, and it comes out as showing an increase in the current. As we add more lactate, we get another stepwise increase, and so on and so forth, and you get the idea. So we get this nice uniform increase. This is actually some of my raw data that I'm showing here, just to show how nice this can look when it's up and running. And from this, we can then calculate how much drain fluid was in the original, how much lactate was in the original drain fluid sample. So I made that sound really easy, but I just wanted to underpin the kind of work that goes into development of a biosensor. So first I tested different types of electrode, and these can be made of a variety of materials that are inert, such as gold and carbon, which we ended up using, to look at which was the best one for our system. One of the key kind of components with biosensing is the attachment of, in my case, an enzyme onto the electrode surface. And again, we tested lots of different ways of doing this to see which was the best one for our system. I also attached different amounts of enzyme for different periods of time, again to optimise it. And lastly, we just added some other substances such as glucose, which would be present in this drain fluid, to check for any additional signal that you're getting, so any interference, which is going to give us a false reading. So once the biosensor is optimised, we then tested it on real drain fluid samples. So I collected 10 patient samples from the hospital here, from one day after the operation. That was after full ethical approval. I tested each of the 10 samples with the biosensor, and all of them also with a commercial kit that we bought in, which takes a number of days and lots of equipment and things to work. So it's not immediate like our biosensor. And we looked at the results. So on the bottom is the amount of lactate the kit said was in each sample, and at the side is the amount of lactate the biosensor said was in each sample. And this is just one patient data, this one here. So the kit said this sample had 1.9 millimoles per litre of lactate in the sample. The biosensor said it was quite a little bit higher at 2.8 millimoles per litre. And the error bars there are the standard deviation, which is just that we measured each test 3 times, and it's a degree of how much those 3 agreed with each other. It's a degree of accuracy. So that's one patient sample. If I add in the other 9, it gets a little bit busier, but essentially if the kit and the biosensor gave identical results for each sample, each patient would lie exactly on the line there. So you can see they don't quite do that, but actually they're pretty close. And for doing it in a lab in a pot, this is actually pretty close data. We applied a statistical package, and this shows that the results are statistically similar, so the biosensor works. We also know that a patient, from the literature, we know that a patient who doesn't leak has an average of 3 millimoles per litre in their drain fluid at this time, and a patient who does leak has an average of 7 millimoles per litre. So already the biosensor would be able to tell us this difference, so it does work clinically as well. So in summary, we've got a working biosensor. It works on real patient drain fluid samples, and it will be able to tell the difference between a leak sample and a non-leak sample, so it will be ready for clinical application. It now requires commercial development. This means making the biosensor with machinery instead of me pipetting in a lab, and this will improve the accuracy and reduce those error bars down. And miniaturisation, so it looks a little bit more like the glucose sensor that I showed you. And the impact of all this is huge. I mean, first and foremost, it's about reducing patient deaths and complications from this joint leak problem that we get with bowel surgery. or with a huge cost saving to the NHS. The kind of technology we use has the potential to be developed for other disease processes, so other joins that we do in various other parts of the body, but also you could measure lots of different markers and look at different disease processes throughout the body, so the potential is quite extensive as well and broad ranging. And it will strengthen collaboration between the NHS and the university. So I'd just like to finish by thanking my funding sponsors and thank you all very much for your time. Thank you.